Welcome everybody to lesson 8 of the metagenomics track of the BVCN, the metagenomics track of the BVCN. It's lesson 8, even though it is the 6th lesson that we'll be putting out, because between the previous one on metagenome assembly and this lesson, there will still be lessons on assembly metrics to check how good your assembly was, and assembly visualization, so looking at your assembly graph. And then binning is something you would do with your assembled contigs. So lesson 8 of metagenomics covers binning. We'll start by going through what binning is and why you would do it. Then we'll go over the principles of how binning works. We'll highlight some of the methods and the tools, and we'll discuss some complementary approaches because that try to achieve the same thing, which is trying to separate your individual organisms out of your complex community data. And then finally, we'll go on and do a little bit of a demo of how the various tools work. All right, so on this slide on the right, you see a principle of what metagenomic binning achieves. It is the left track on the diagram, and the colored circles up top represent a complex microbial community that consists of different species indicated by the colors. Now, if you extract DNA from that community and then sequence it and assemble it, as we've discussed in previous lessons, what you'll get is a mixed bag of longer stretches of DNA called contigs that represent all the members of the community mixed. However, in the environment, the organisms and their genetic material exist in discrete cells. So to correctly interpret the genetic information of the environment, it is important to try and separate out the individual, the genetic potential of the individual organisms in the environment. This separating out of contigs in a metagenome is what we refer to as binning. And, well, yeah, there we go. It is currently largely restricted to bacteria and archaea. And this is mostly due to the streamlined genomes of bacteria and archaea and the fact that their genome sizes are much smaller than those of eukaryotes. This means that typically DNA from bacteria and archaea assembles better into larger and longer contigs. And we'll see later why that is advantageous for binning. And because their genomes are also much smaller, less sequencing is needed to get to a sufficient coverage, so sufficient abundance in different samples to inform the various binning methods. It says on the slide it's possibly a transient problem. The reason for this is that as sequencing quality, and especially long-read sequencing quality, improves, eventually we might get to a situation where we can resolve individual, and I'm going to say microbial genomes in single contigs. And if the entire genetic potential of an organism is covered in a single contig, it is already binned. There's no more necessity to bin it. By the time we reach that, eukaryotic binning will also be like a much more prevalent thing. And that will open up a whole new world of studying eukaryotes in the same way that it's done for bacteria. So why do we do binning? One of the things that has been proven over the last decade or so is that it is a fantastic way to discover novel organisms. In a similar way that in the 90s and maybe early 2000s, PCR revolutionized our understanding of microbial diversity and of the tree of life, binning has added an extra layer to this. The reason for that is that it it allows us to discover organisms in a reference-independent way. Whereas PCR, you require a little bit of knowledge on what the 16S of your target organism looks like. Binning is fully reference-independent and is only dependent on biases in sampling or in how your processing pipeline affects the DNA of different organisms different. Now, once you have discovered novel organisms, you can look at where they sit in the tree of life. And one of the prominent examples of how this shaped our insight in what the tree of life actually looks like is the Asgard archaea, the group that is currently thought to be basal to all eukaryotes and our closest archaeal ancestor, so to say, and has firmly established the tree of life as a 2-domain tree rather than an actual 3-domain tree. The other thing, trait evolution, has been a big thing in methane and alkane metabolism. We thought initially that methane production and oxidation could purely be done by a group of organisms in the Euryarchaeota, but with more and more genomic data from the environment, we found that these sequences exist all across the archaeal tree of life. So we now have a much more comprehensive view of how this trait evolved through the archaea. I say predicting community interactions, this one is contentious. To a certain extent, if organisms co-vary through multiple samples, you might be able to predict community interactions from them. One of the prominent examples there could be these CPR organisms, or the DPAN archaea, which look like they are ectosymbionts on larger bacteria or archaea. And sometimes hosts can be predicted through their interaction with other organisms. And then finally, hypothesis generation on what all these novel organisms do. This is a big one, and this is a big one that people have speculate on a lot, and that requires a lot of experimental verification, which is contingent on culturing most of the organisms that we find. And this is still one of the major challenges in microbiology. So in a nutshell, how does it work? So it's really a very simple process that can be implemented in many different ways. But in a nutshell, there isn't a whole lot of information in DNA. That is not, well, obviously there's a lot of information in DNA, but there isn't a whole lot of information beyond what it codes for. So all we have to work with is the composition, which is a conserved feature across microbial genomes, the abundance of the DNA across several samples, and the taxonomic affiliation of the encoded features. So an example of that, what that would look like, is shown on the right. This is a plot from Mats Albertsen's 2013 paper, where he took all the contigs in his metagenome assembly, and this was a metagenome sequence based on 2 DNA extractions, one without a hot phenol treatment in the DNA extraction, and one with the hot phenol treatment. The without is the x-axis, the with is the y-axis. And you see that the hot phenol extraction differentially favors certain organisms with mostly high GC content. The axes represent the abundance of each scaffold in each sample. So you can see that clusters with similar GC content and with similar abundance form fairly readily. You can then annotate those with their taxonomic affiliation by predicting the features on the scaffolds and seeing where they which other organisms they match. And that way you get these emergent clusters. And you'll see that something like bin 13 in the top right is going to be much easier to separate from the other organisms than bin 22 in the bottom left, which is fully overlaid with a different one. So in a nutshell, this is a callback to lesson 2, where Eric mentioned that the sequence composition is conserved across the genome, and therefore we can use it in binning of or in grouping of contigs or scaffolds together. This slide from a 1998 paper also shows the one of the issues with that. There can be genomic islands, like the one shown by the black bar on the right-hand side of the figure. That is, that have different composition and therefore might not group with the rest of the genome, even though they clearly do belong there. A notable problem with this is the ribosomal operon, which is much more constrained in its sequence composition than any other part of the genome, and always sits between 40 and 55% GC, regardless of GC content of the rest of the genome. Then genome coverage is proportional to organism abundance. This is not entirely true, because there is a bias. There's biases in DNA extraction, for example, and might be some biases in processing. But to an extent, the abundant species in your sample will be abundantly present in your DNA. Therefore, will have a higher coverage. So if you have multiple samples, you can then use the coverage pattern across, the abundance pattern across samples to try and see which contexts group together over multiple samples. This is called differential coverage or differential abundance binning. In a nutshell, in practice, what this can look like is something like this. The reactor on the right-hand side here shows what is technically a metagenome. It's about 95% one organism, but this class of organisms, Anamox bacteria, cannot be grown in pure culture. So to extract to obtain a genome of an Anamox bacteria, you always have to go through metagenome binning. And as you can see, this truly is the best case scenario for metagenome binning, where on the left-hand side on the plot, each contig represents a dot, or each dot represents a contig, and the size of the dot is proportional to the length of the contig. And we see there's one cluster of contigs in the middle that represents the genome of the dominant Anamox bacteria, and then this smear at the bottom that represents any other organisms that are there, but there isn't enough DNA to resolve any clear clusters. What you also see is that there's this halo or cloud of contigs around this. These very likely belong to the Anamox genome, but for some reason have a diverging coverage. These things at the top may be duplicated. The ones at the bottom may be mobile genetic elements or sorts. Now, if things get a little bit more complicated in a different Anamox reactor, we have two organisms that cannot be easily separated by their GC content and have overlapping coverage. So now our compositional and single abundance binning is no longer enough to separate these two organisms. So the way we'll go about this is, rather than using the abundance in a single reactor, we'll use the abundance in two different reactors. And what that looks like is like this. What we see here is that the y-axis in both plots is the same, but the x-axis now has the coverage in one reactor instead of the GC content. And we see that the two abundant organisms separate out by their coverage across two reactors. What is also of note in this plot is that over here in the right plot, at the top, we see just above the blue circle, we see a cloud of small dots that is that has higher coverage than the red genome, but clearly belongs to the blue genome. The reason I say this is of note is in this organism, several of the key genes for its metabolism are duplicated. These will, due to assembly chimera protection, be on their own contig with double coverage, and therefore typically not bind with the organism in question. So in this case, one of the key enzymes in metabolism is on one of these clouded dots. So even if you have a good quality genome, you might still be missing essential bits because they are duplicated. This is another thing where the 16S suffers, or the ribosomal operon suffers. This is often present in more than one copy, and therefore will have different abundance than the rest of the genome and is likely to get lost. So going into the various approaches that people have taken for binning, there is the set of manual approaches, which can be used if you have, or which should be used if you have one or a few genomes that you really care about. The reason I say this is the manual approaches give you a way of specifically making the decision for each contig or for each genome, and tailor the decisions that you make to each genome. For example, in the previous plot, in the manual binning, we can include these things, the contigs above the blue circle, into the blue genome because we are fairly confident that they belong there. In automated binning, these would likely not be detected. There's a bunch of approaches for manual binning. One is called Genome, which is the plot we saw earlier, and what the plots in the previous two slides were based off of. A big one that a lot of people use is Anvio. This was originally developed as a binning and visualization platform, but is now sprawled into this multifunctional bioinformatic powerhouse, and I encourage everyone to try and learn it. We will cover it in much more detail in a later lesson in metagenomics. ESOM, now ESOM is short for Emergent Self-Organizing Maps, and is a machine learning algorithm based on sequence composition, and can be used to delineate organisms in a not so complex community. It works relatively well, but has largely been superseded by other approaches. And then there's other purely composition-based manual approaches that make a matrix and then do dimensionality reduction and clustering on that matrix. The big advantage of these things is you can really tailor and choose what you want to include in your bin. The big disadvantage is it's really low throughput, and so for many of the larger analyses that happen these days, it is not feasible to do this manually. The other thing that is also a big drawback is that because the reproducibility of your binning strongly depends on how well you document what your choices are. And that has been a problem in the past and is somewhat impossible with ESOM. Now then we get to automated binning approaches. Like starting from 2014 on, after differential coverage was became accepted as a way of, or as a method in separating contigs belonging to different organisms, a lot of manual binners sprung up quickly. They're relatively easy to run, although they do have some like very specific requirements that you have to be aware of, which we'll cover in the second part of this. They use the same underlying information as manual binning, which is the abundance across samples, the coverage, and sometimes the taxonomic affiliation. All of these all binners use that same information, but have different prioritization of that information and have different weighing of how important each component is. It's in these things often the case, the more samples, the better. So if you find yourself in the project design stage of a metagenome project, try to arrange such that you have a library that is split along a time series or multiple samples from similar but related locations, or even different DNA extractions from the same sample, so that you can get, or another way of splitting the sample, so that you can get leverage that differential abundance across your samples into the binning of your context later. It is thinking about this upfront in your design, in your experimental design, is really important to get this. It's really important to get like the most out of your data later. There's been a couple of large-scale evaluations of these things in the context of the critical assessment of metagenome interpretation, CAMI challenge. And one of these assessments is shown on the right. What we see is that what we see is that there's a large heterogeneity in the quality of the bins that are produced. Right, what these box plots on the right show, at the top is the purity per bin, so how clean is the bin, and the completeness per genome. So what we see here is that both MetaBot and Binsanity do very well. Binsanity with the workflow do very well on getting largely complete genomes that are also of fairly high purity. The tool at the bottom outperforms any individual tool, but is a binning aggregator. We'll talk about that in a little bit. So from here, we should go and briefly discuss the possible errors that you get in binning. These are very well represented in the graph here from Meyer et al. 2018. On the left-hand side here, we see the actual context that grouped with the organisms that they belong to. On the right-hand side, we see the result of a binning effort. So in green, things are the true positives are represented. In red, false negatives are represented, so things that actually belong in the bin but have not been grouped with the bins. And in blue, false positives have been. So things that are grouped in the bin but don't actually belong there are indicated. The shape of each of these dots tells you which bin they actually belong to. Then the formula at the bottom shows the formula for how Meyer et al. Calculates the adjusted RAND index, which is a measure of how many true positives and false positives there are, true positives and false and true negatives there are over all true positives, true negatives, false positives, and false negatives, and then with an adjustment factor. This is a way of evaluating how well the bins have done. And this could be done by the CAMI challenge because they had a gold standard, so the actual real answer. These are two plots that show that adjusted RAND index that we just talked about in the previous plot. And from the CAMI challenge itself, and then from the assessment of metagenome binning, I forget what the AMBER acronym stands for. So we see that there's a few more. The AMBER paper supersedes the original CAMI paper when it comes to binning. It has included a few more of the recent binners. Notably, it has included the second version of MetaBot, which outperformed. This is the first version, as was in CAMI, and then the second version is over here, so clearly outperforms the original version. And it included Binsanity, which is a more recent tool written by, amongst others, Ben Tully, who spearheads the BVCN. In these plots, being as far to the right as possible tells you how accurate your bins are, and being as far to the top as possible tells you how well you do in binning all of the contigs in the data, or many contigs in the data. We see that max bin, by the standard of correctness, so is the best binner, but is very conservative in what it includes and may therefore have lower completeness bins. We see that in both plots. The DAS tool software, oh sorry, there is no DAS tool software in this plot. The DAS tool software in this plot outperforms all of the individual binners because it takes the best, or it tries to take the best bins from all binners used as input. So we'll cover a few more in some detail. We'll cover Metagenomics, and we'll cover Binsanity, and we'll cover DAS tool. Because they are both commonly used by people in the community, and they're generally good for automated binners. So Metagenomics uses tetranucleotide frequency, so sequence composition, and coverage across samples, and uses that in a way, uses that to assign a score based on the abundance and the tetranucleotide frequency, and then initially builds a graph of all the contigs, not based on this score, but based on the matrix of tetranucleotide frequencies. So a tetranucleotide frequency, to recap, is when you take a contig or any stretch of DNA and calculate all the possible 4 nucleotide sequences that are present on that stretch of DNA. So base 1 to 4, base 2 to 5, base 3 to 6, etc., etc. And then count how frequently every of those formers, or what the frequency of every possible former is, tetramer, is on that contig. This gives a decent, this will group contigs that are similar and split contigs apart that are dissimilar. So Metagenomics, importantly, uses sequence composition as its starting principle to build a graph to initially cluster things together. Then it will use a label propagation algorithm to partition this tetranucleotide frequency graph, and uses the score that is shown here as a quality, or as input in this label propagation algorithm. So it is initially based on composition, and then secondarily on the abundance. Binsanity takes the radically opposite approach. It is fully based on coverage, with then subsequent refinement based on tetranucleotide frequency. So differences between Binsanity and Metagenomics could arise from the fact that they take their primary, or use a different aspect of the similarity between contigs to do their initial binning. And it's not that one is necessarily better than the other, because let's say if we have an organism with 60% GC in a community where all the rest have 40% GC, then sequence composition is going to really beautifully resolve that organism. Whereas in a community where you have an organism that is 80% of the community, abundance will really beautifully resolve that organism. So different scenarios might favor different binning approaches. So the graph on the right is from the Binsanity paper, and basically shows the workflow for Binsanity. So it starts by making an alignment file, then a coverage profile, merge all the profiles into a matrix of all the coverage, all the contigs and then the abundance per sample for each config. And then cluster that using affinity propagation, which tests each individual config as the center of a possible cluster, and then picks the best centers and clusters based on that. Then bins get quality evaluated using the Check'em software. We'll cover that separately in a different lesson. And then compositional data is used to refine the bins if needed. So the last automatic algorithm we'll cover is the dust-tool algorithm. Dust-tool is a binning aggregator. It's the only one. And what it does is it takes the bins from different algorithms. So what you do is you run your assembly through several different binning softwares, such as Binsanity or Metabot, or looking at them manually. You can take any of these binning outputs as input. What dust-tool will do is it will assign each of the bins across your assembly a score. And for this, it's important to realize that the graph on the right-hand side shows bins in colored ellipses, and then contigs in black stripes. The contigs are overlapping between different bins, right? There's one assembly that is then partitioned based on different techniques or tools. So that means that the contigs in each bin are overlapping, or contigs between different binning algorithms can be overlapping, but the bins are separate from one another. So it scores. So what it then does is it scores each of these based on single copy marker genes. This is a way of assessing how good a bin is based on the presence of genes that we expect once and only once in the genome. These single copy marker genes will then be scored based on their number of unique single copy marker genes, USCG, divided by the number of redundant single copy marker genes. Say there is 100 single copy marker genes. I forget exactly how many dust-tool uses. And you have 100 unique ones, or you have 95 unique ones and 5 redundant ones. That means that you have some contamination. Then the second term is a penalty based on the duplicated number of single copy marker genes. No, I'm not saying this right. The first term, unique single copy marker gene divided by the total possible single copy marker genes. So that is purely a completeness term. So 95 genes out of 100 possible gives you 95% completeness. The second term is a penalty for duplication. Duplicated single copy marker genes divided by unique, so how many genes are present twice, as a fraction of how many are actually present. And then the third term is what they call a penalty for mega bins, where bins are really large bins that consist of, say, multiple genomes. This is a common feature happening when genomes are closely related taxonomically. They probably have a closely related GC content and fairly equal abundance. You'll get lots of overlapping bin content. So the third term is the total single copy marker genes in a bin minus the unique ones divided by how many there should be. Now what it first does, as visually represented on the right, is it takes these single copy marker genes, assess which bins overlap, and then picks a bin based on the score. If the score is the same, say if both of these have the exact same set of single copy marker genes in them, very likely, or somewhat likely, it will use the N50, which is an assembly metric which is how it's the length, no, the number of contigs that the minimum number of contigs representing 50% of the total assembly length is what N50 is. So say you have 300 contigs and you organize them by length. Your 300 contigs are a total of 2 million base pairs. Then the N50 is the number of contigs, minimum number of contigs you need to get to 1 million base pairs. It basically tells you how connected your assembly is. And then if that is also the same, it uses total length of the bin. So the first one picks content, the second one selects for large contigs in the bin, and the third one is inclusive of all the small, any small contigs that are still included in the bin. What it then does is it takes these, it resolves the differences between the bins as depicted here on the right, and it takes a new bin set, and then it repeats. Keeps doing that until there isn't any differences to be resolved anymore. This is a different way of evaluating bins. This is from a blog post recently put forward by Miran, who is the developer of Anvio. So he has some sort of an agenda here, but is generally very invested in making sure that these things are done well. What this shows is, God, what's the name of these flow diagrams? These diagrams, the one on the left and the one on the top right, track the fate of each contig across the different binning tools. So it's a visual way of seeing how DustTool operates and how different binning tools operate relative to one another. Fluvial diagrams, I guess. So here we see that there's one bin, one very large bin of concoct, which is 100% complete, but also a lot contaminated. Complete here in green, contamination in red. We see that Max bin disagrees and says, no, these are 2 bins. And then there's a whole lot of contigs that are present in, that are not either not binned or in different bins, in the case of Max bin. Metabot does something analogous, but says, no, there's 3 bins. And there's a bunch of smaller contig, or small components that get placed in different bins. DustTool then, based on its aggregation and recombining method, first picks the Metabot bin from here, and then combines the remaining bits from the concoct bin and puts them in a second bin. So both of them have now a decent completeness, although the top one here is fairly contaminated. So you see there can be quite big differences between how the various binners group their contigs. Then his final point with this is, even though DustTool in some cases does a very, very good job and does better than any of the individual binners as we've seen in the metrics previously, we can still improve upon it sometimes by looking at the bins through the lens of a manual inspection. This is what's shown here on the top right. On the top right, what we see is the same plot as we see in the left, but there's one extra line added, which says manual. The manual line is colored green and purple, and those green and purple lines correspond to the green and purple line at the bottom plot. We see that relative to DustTool, when we look at the Anvio plot at the bottom, we see that in some of the samples, the outer ring is relatively dark colored, here in green and not so much in purple. So we see that by looking at it manually, even though the sample that drives both of these bins, the abundance is relative. The 3 samples that drive both of these bins, the abundance indicated by lots of dark coloring here is similar. Their abundance across these two samples, like on the outer layer here, is very different and separates them out in indeed two different groups, even though this would be a harder call to make. But because Unreal calculates things in a slightly different way, it separates them out relatively cleanly. So there is, so that gives us a possible view of how manual refinement still has a place in looking at bins, even after you've manually refined them with dust tool. So it is important to inspect these things and assess. In this case, it can be questionable what this area over here in roughly the middle of the green bin that seems to be, while the abundance in these three samples at the inner ring agrees relatively well, there seems to be differential abundance. So this might be like a mobile element of some sort. So that brings us to other methods that try and achieve the same thing. So taking a complex community and separating them out. One of them, the big one, is single-cell genomics. Right? And in the figure on the right, which we saw in the beginning of the presentation, I've juxtaposed the metagenomic binning and single-cell approaches. And the big difference with single-cell, obviously, is that it does separation first. Right? Metagenomic binning does the whole process and then separation last, whereas single-cell takes your community, separates out the cells of interest, or separates out individual cells, and then using genome amplification and sequencing and assembly, you can get to a draft genome. We will not get into it here because single-cell genomics requires its whole own module even. But briefly, the big pro of single-cell genomics is you can start doing microbial population genetics, right? Because you get the genotype of an individual cell. Whereas in metagenomics, strain heterogeneity often gets lost. The big, and it is possible to pick up rare cells from the environment that would be really hard to resolve in metagenome binning. The cons is because of the genome amplification, the multiple displacement amplification, completeness used to be really bad and is still often slightly worse than from metagenomics, but is rapidly improving with improvements in chemistry. It is a little bit more intensive in the sample prep and is slightly lower throughput than metagenomics. But we won't go into it further here. Then another complementary method is high-C metagenomics. This is a trick where you cross-link the DNA replicons in the cells before you lyse. So this is depicted here on the right. Say this budding yeast in green has two chromosomes, right? Two linear chromosomes depicted as these green lines in the budding yeast. What high-C metagenomics does is it takes, it adds, I believe it's formaldehyde, to the cells, fixes them by cross-linking randomly. Then while the DNA is still cross-linked, you lyse the cells, take the DNA out, and digest the DNA. So basically, using restriction enzymes, cut it in a bunch of different places. Blunt end restriction enzymes. Blunt end because that means they can randomly religate. That is the next step. You randomly religate, so you get a completely scrambled, discombobulated genome. The idea is that because everything is cross-linked, this is a fixed blob where you've made some cuts. The random ligation has to happen close to the physical distance between the two bits that get randomly ligated to one another has to be small, right? So then what you do is you sequence both a native library where you haven't done any of this to actually get the proper gene information, and you sequence your cross-linked library. And then you use the links in your cross-linked library to infer what is close together in like, and here at the bottom, what this shows is warmer colors tell you that things are very close together, or tell you that things are linked a lot, and colder colors tell you that there's not so many links. So there's always a little bit of links that are spurious. But here, for example, we can see across the center line that DNA from the individual replicons has the most linkage to itself. What we also see is that this Burkholderia organism, which is a microbe that has 2 replicons, its 2 chromosomes have more links within themselves than between them, but they still have many more links between them than to any of the other samples in the community. Then these 2 E. coli's, which only have 1 replicon, are very well separated, so it doesn't matter that things are phylogenetically closely related. These are the same, different strains of the same species, and you can separate them. And then here there's a lactobacillus with 2 plasmids that again can also be separated, like can also be grouped together. And a little bit, the chromosome is a little harder to separate from the plasmids in this case, but it is it's more lab intensive, but it is an interesting technique to apply for things like viral infection or parasites or other things that are, or plasmids. You can resolve plasmids in a genome, which otherwise often get lost. So it gives you extra information you wouldn't get from a regular metagenome and binning workflow. Another analogous method is called read partitioning, or termed read partitioning, which is using, rather than, it is a binning method in a way, but rather than binning contigs, so long assembled sequences with a lot of information in them, what you do is you partition reads. So you try to group reads together. This was the original binning. So the first binning tools tended to rely on this method, which uses long k-mers, so say 33 or 35 nucleotides, and the assumption that each k-mer is unique in a genome. And then as you walk across a read and encounter a bunch of these k-mers, you can group the reads together based on their long k-mer content. This has largely been superseded by contig binning, but might still have a place in places where assembly is really, really challenging because of computational resources. You can limit the amount of memory that you need, for example. So the graph on the right is from the latent strain analysis paper from 2015, made a big step in this type of tool. And this has been revived once more in a paper that came out about a month ago the paper that came out about a month ago showed that there is that this type of read partitioning, when done well, can separate out or can resolve individual strains, which is a big problem in metagenomic sequencing, and in theory at least, and it can and the bins that you recover are not necessarily overlapping with the bins that can be recovered with assembly-based binning. It is better at recovering really low abundant organisms. So there might be a place in a parallel workflow for something like this. Again, this does require that you have the more samples you have, the better this works. Then finally, this is a potentially emergent tool or technique, and would have been best to be introduced after the assembly visualization lecture, but still, this is graph partitioning. So rather than what happens after assembly, where you take a graph, where you get a graph, rather than taking that assembly and extracting DNA sequences from it, or taking that graph and extracting the most probable DNA sequences from it, you leave the graph with all its imperfections and weirdnesses and possible sequencing errors. And you use that as the source of your data. There is currently one implementation that I know of, software called Spacecraft Cats, currently in preprint. And it would allow for, one of the big things here, this would allow for fully understanding the community complexity at a strain level. The images on the right show a part of how that graph partitioning would work. At the top and at the bottom, what it shows is representations of the graph. And the insets, the dot plot insets, show variable amino acid sequences as assembled by an amino acid assembler. And what you see on the graph pictures, so top right, middle left, bottom right, is that there is a lot of these loose ends. When you go from your graph, like here, for example, in the bottom, you go from your graph to your sequence, to your context sequence, these loose ends get resolved, or these bubbles and loose ends get resolved, which means you pick one and the rest gets lost. And what Brown et al. Showed is that if you look at the graph, you can still get a lot of variation that is inherent, or that is present in the graph but lost in the context, and you can use that in your analysis. And with that, I am going to end it for today, and we will go and move forward with the demo. But first, if there are any questions, I will happily take them.